So the question is that given that the major neurodegenerative diseases are largely caused and maintained by the misfolding of normal brain proteins, what is Amprion's role in helping to detect and manage these diseases more effectively? And that role includes helping patients and their doctors understand who has which molecular defect, and helping drug companies to target those specific defects for the next generation of effective drugs. So just to recap, Alzheimer's appears to be caused by misfolding of two predominant brain proteins. One is Abeta amyloid, one is tau, and Abeta and tau both can misfold in several different ways. So we have the normal protein, we have a misfolded form that leads to very large insoluble aggregates, and we have a second misfolded form that leads to soluble small aggregates that appear to be able to spread, and this is probably the carrier of the disease in the brain. So in general, the normal forms of these proteins can be detected by very standard methodologies. However, the normal form doesn't reflect the disease. The misfolded form in this form of large soluble aggregates can be detected initially under the microscope, but since we would like to avoid doing biopsies for brain, they can also be detected more and more by imaging. However, these large insoluble aggregates do not appear to be linked so tightly with the disease. There are many patients with Alzheimer's that don't seem to have plaques or tangles, and there are many patients who are normal aging who have plaque and tangle and don't appear to have disease. So, Amprion focuses on the small, soluble, misfolded aggregates that many people believe are the actual carrier of the disease. Amprion can uniquely detect these small soluble aggregates. We can detect them in the cerebrospinal fluid. We are working to be able to detect them in the blood, and we believe that this is the hallmark of the disease. This is the carrier of the disease. This is what spreads it between cells without knowing about how much and what the structure are of these small soluble aggregates. It's very difficult to have a handle on the disease. And I think it's fair to say that if you only look at the normal and the large clumps, that doesn't give a sufficient picture to be able to understand what's going on in a patient. However, looking at the small soluble aggregates appears to be necessary, and maybe necessary and sufficient to diagnose and treat that disease.